Hello, fourth period. Hopefully you are finding this video on YouTube. No big deal. This is going to be the replacement lecture for October 13th since we have advisory that day and we don't have time in class to do the lecture. So hopefully it's not too terrible for you. So we're talking about the evolution of populations, chapter 16 out of the textbook. I highly encourage you to make sure that you have taken the notes in your notebook already before watching this video, so that way you can just go through the video and highlight the words as we move along. All right, so getting started, we're talking about Darwin again. Now remember, Darwin said one of the most important principles of natural selection is that each population has variations. So he felt that that was very important, but he did not understand how variations were passed from parent to offspring. The funny thing is, at the same time, there was a monk named Gregor Mendel, remember him from genetics, who was studying pea plants, and he figured out that there was a factor that was being passed from parent to offspring that was helping the parents pass their traits on. Darwin and Mendel lived at the same time, but they didn't know anything about each other, and it wasn't until the 1930s that people started linking their work together and kind of gave us an idea of how natural selection works, excuse me, on the variation in a population. So let's talk a little bit about variation. So many genes, as we know, have at least two forms. So think about the pea plants that Mendel was studying. They could be tall or they could be short. So there's a gene for tall and there is a gene for short. So these two different forms of the same gene for height are called alleles. Now the genes that you get determine your genotype. And we have a couple of words we use to describe genotype. Now remember, the prefix homo means the same. So if we say an organism is homozygous, that means both of their alleles are the same. If they get two dominant alleles, which are represented by capital letters, we say they are homozygous dominant. If they get two recessive alleles, which are represented by little letters, then we say they are homozygous recessive. Now they can get one of each. They can get a dominant and a recessive, so we call these guys heterozygous. Now, in addition to describing their genes, we can also describe what they look like, their physical appearance, and we refer to this as the phenotype of the organism. If I was to get all of the alleles and genes in a population and get them all together, that would be our gene pool. And the number of times a particular allele appears in our gene pool is called the frequency. So we've said before that evolution is basically change over time. We can also say that evolution is any time the frequency of alleles in a population are changing. Now, when we talk about the differences between us, the sources of genetic variation, there are two main ways we get this variation. The first way is through sexual reproduction. Now, think about it. You do not look exactly like your father or your mother, because you only have half of your alleles from your dad, or half of your dad's alleles and half of your mom's alleles. So this means that you are a completely unique individual, unless of course you're an identical twin. So sexual reproduction ensures that each individual in a population is a little bit different than every other individual. The other way we get genetic variation in a population is through mutations, random changes in DNA. So think back to the rock pocket mouse. Remember, the black coloration is a mutation. It's a random appearance. And mutations can lead to changes in phenotype, like with the black mouse. Mutations can be good or bad, or they can be neutral. It just depends on what's happening. Does it increase the fitness or does it decrease the fitness? Or maybe it has no effect. Now there's this idea in biology, which we call the central dogma, which basically says that DNA codes for proteins, and the proteins determine phenotype. So basically your DNA determines what you look like. Now there are two different traits we're going to be focused on here for this particular set of notes. The first one is what we call a single gene trait. So pea plants, being tall or short, would be an example of a single gene trait. There's only two alleles. We also refer to this as a discrete trait. Another example of this would be widow's peak. So if you look in the bottom left-hand corner here, you see Thor. He has a widow's peak. It's a dominant trait. You either have it or you don't. When I represent, or when I show single gene traits using a graph, I use a bar graph because there's only two phenotypes. The second type of trait we're looking at is a polygenic trait. This is a trait that is controlled by two or more genes. We call it continuous. An example of this is height in humans. So if we were to get the height of everybody in Mayfield and map it out or graph it out, we would come up with a bell-shaped curve where the majority of people would fall in the middle, would be average. You'd have some really short people and some really tall people. So it's really easy to tell the difference between these two when they're graphed, because in a single gene trait, you only have two choices, whereas in a polygenic trait, it's on a continuum. It's kind of like there's a lot of different choices. We see the same thing in human skin color. Now, when we talk about evolution, natural selection doesn't act on the genes. Rather, it's affecting which individuals survive or die. So it's acting on the phenotypes, which are, of course, determined by the genotypes. So when we talk about single gene traits, natural selection is pretty straightforward. It can change gene frequency pretty easy. So if we think about the rock pocket mouse, we have that black mutation show up on the lava rock, and then we see evolution really push the population towards all black colored mice. Same thing in the antibiotic resistance lab. In a polygenic trait, you can have three things happen. The first thing is what we call directional selection. So if you look at the bottom left, you see this blue kind of bell curve. On the left is going to be the beginning. And so in this bell curve, you'll notice you have the same as height in humans. You have most people or most individuals in the middle, in the average, and you have some extremes. In directional selection, one of the extremes is favored. So you actually see the population move in a direction. We're kind of pushing it. You can also see this in the middle picture with the horse. At the beginning, you see this tiny dog-sized horse, but the selected phenotype is going to be in blue, and we see it move over time to where we now have a much larger modern horse. The second type of selection is called stabilizing selection, and this is where the average individual or the individual in the center of the curve have a higher fitness. So we see that kind of in the middle part B on the picture on the left. The last one is disruptive selection, and it's hard to see. It's probably cut off on your picture. But basically what it's saying is that individuals on both extremes of the curve are selected for, and the average individual selected against. So we actually disrupt that curve, and we see an increase in both extremes. All right, there are other ways to change gene frequency rather than natural selection, and the first way is what we call genetic drift. So imagine we are a population of bunnies in Kansas, and we all have variation. We're all a little bit different, and a tornado comes and wipes out half of the population. Well, those of us who survived not because we were more fit, but we just happened to be in the right place at the right time. So we've changed the gene frequency, but it's due to chance, not to natural selection. And then the last one is founder effect speciation. Basically, a population has a change in gene frequency because of migration. So a group of individuals starts a new population. Because they've left a bigger group, this changes our gene frequency. This can also lead to speciation, or what we call the formation of a new species. So how can we tell if a population is not evolving? That means it's at equilibrium. Well, two guys actually came up with this idea on their own. Hardy and Weinberg came up with this theory called Hardy-Weinberg equilibrium. And it says that allele frequencies in a population will remain constant unless one or more factors cause them to change. Now, there are 5 conditions a population has to meet in order to be in Hardy-Weinberg equilibrium. There has to be random mating, so no sexual selection. It has to be a very large population. No immigration, so basically nobody comes in or out, no mutations, and no natural selection. There are 2 equations we're concerned with, P plus Q equals 1, and P squared plus 2P squared plus 2PQ plus Q squared equals 1. I'm going to go over this more in depth on Friday. So how do new species arise? First off, that word species should be underlined. I forgot to underline it, so make sure you highlight it on your notes. So a species is a group of organisms that interbreed and have fertile offspring, and that's the key there. They have to have fertile offspring. Now, if we think about a horse and a donkey, they are not the same species, but they can get together and have an offspring, a mule, but that mule typically is infertile, so it does not count as a species. That fertile offspring thing is really important. And if we have a lot of small changes that lead to new species, we call all those small changes microevolution. So in order for a new species to arise, then the population must become reproductively isolated. And I'm realizing now as I'm looking at this, I didn't highlight these words. So reproductive isolation, behavioral, geographic, temporal, mechanical, and gamete all need to be highlighted on your notes. I apologize. So in order For a population to be reproductively isolated, that means that they can't interbreed. So in here, behavioral isolation, two populations cannot interbreed and produce fertile offspring because their behavior is keeping them apart. So the example we have here is a blue-footed booby. The blue-footed booby finds a mate by waving its feet around and doing this funny little dance. There's a link to a video under modules. You can look at that. There's also a red-footed booby, and it does a different dance. So these two boobies are very closely related, but because of their different behaviors, they tend not to reproduce. We also have geographic isolation, which is pretty simple. Two populations are kept apart by some sort of barrier, like a river or a mountain. Temporal isolation is when two species are closely related but breed at different times, so they tend to be isolated that way. Mechanical isolation, I'm going to go ahead and switch to the next one so we can see the picture. In mechanical isolation, the sex organs are not compatible. So most birds don't have a penis, but there are some exceptions. One of those is a duck, which has a corkscrew penis, as you can see in this picture. The female ducks have corkscrew vaginas. What happened here is that male ducks tend to force copulation on females. Females don't particularly like this. They've evolved a vagina so that if they are being forced to have sex against their will, they will actually, if you can see in the picture, the penis will come into a little pocket that doesn't lead to the ovary, and so it doesn't fertilize the eggs. If the female is mating with a duck that she wants to mate with, she will maneuver so that the penis can actually travel all the way into the right place and fertilize the eggs. And then the last one, gamete isolation. In sea urchins, there are many different species. If their eggs and sperm meet, their gametes will not fuse and form zygotes. So this keeps them reproductively isolated from other sea urchins. So hopefully everybody was able to make it through this video. It looks like we're at 12 minutes. We will be studying Hardy-Weinberg more in depth in class on Friday. Thanks and have a good day.